So let's say you found a skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
All right, everybody. We are here with Alex Ruebenstahl, and I have to say first, I'm sorry, but apparently my internet is just kind of being crap today. I don't know why. I'm trying to get rid of any other things that might be using my internet and sucking up bandwidth, but I don't know. So bear with me. Sorry about that. My OBS is saying that I'm dropping more than half of my frames, so hope that this is actually going to be bearable. Let me know in the chat if this is working. But yeah. So Alex, let's let everyone know who you are and yeah, why you're here to talk about paleontology.
Sure thing. So I'm Alex Ruebenstahl. I am a second-year PhD student at Yale, vertebrate paleontology. I did my undergraduate and my master's degree at George Washington, D.C., and a lot of that work I did was with early crocodile evolution. Currently, at Yale, I'm continuing some of that work in the broader context of some questions about archosaur evolution. So I'm looking a lot at crocodile evolution, as well as pterosaurs, a little bit, not a lot, and then dinosaurs, mostly theropods, and birds. So kind of on the line to birds. So
okay,
that's currently what I'm doing.
Awesome. And so a lot of people probably don't know this, but you're the one who gave me a lot of debate prep coaching for my debate with Kent Hovind.
Yes.
Yeah. So you looked over my my script for my initial opening comments. You looked over my slideshow. You gave me some extra notes, and so I wanted to say again to you, because I've already said it. In fact, this is actually your second time on the channel, but I just wanted to say thank you again because that was extremely helpful.
Of course.
So yeah. So basically, I guess the first thing I want to ask is like, so what's new in paleontology? What cool things have people not heard about?
Oh. So much. So it's actually today is the last day of the Society of Vertebrate Paleontology's annual meeting. They meet every year. And
David Peters is not invited.
No, David Peters is not invited. Although probably the first bit of news is funny news that has very little to do with science. But so the convention was supposed to be in Cincinnati this year. And a lot of us were looking forward to that because we were going to go to the Ark Encounter. It's like big way. But they're doing Cincinnati in 2023 now, so it's fine. But they did it online, and they used this kind of business meeting software to host it, and they didn't know it came with these basically it censors certain words. And it censors hell, so Hell Creek.
Oops.
It censors bone. So for a paleontology conference, that's not great. They sorted it out within the first day, but for that first day, it was very funny. You can't say it. For some reason censors stream, stroke, anything that could even possibly be recontextualized.
That's pretty heavy censorship right there. That's
yeah. I think they sorted it out, but I saw like an article in like I think the Guardian had a piece on it. I was like, oh, no way. How did this make it to a news site? This seemed like such a cool thing. But yeah, so, I mean, that was funny. There's a ton of new research at SVP. It wasn't as well attended as the one in Albuquerque 2 years ago, which was the last one I was at. But unfortunately, I mean, a lot of that work I can't share because it's
not quite published.
Not quite published. But there are some, general trends in paleontology that were pretty common throughout that talk that I can touch on and that I do a little work on myself. So probably one of the most exciting, in my opinion, of the most exciting kind of emerging fields in paleontology, at least in vertebrate paleontology right now, are the use of CT scans and some taphonomy questions. So if you're not familiar with a medical CT scanner, essentially you're putting sometimes a person if it's a big one, or an object if you're looking at a smaller object in a micro CT scanner into this. It takes a bunch of pictures in all orientations, which we call slices.
Mm.
And then you can use software to take those slices and reconstruct 3D images of not only the exterior of the skull, but they scan the inside of the skull too, so you get internal structures.
Yeah, it's that way we can non-destructively look at the entire structure of the fossil, right? Which is that whole non-destructive thing is key, because actually I was at a museum with my nieces, and they have a little section where you can see people doing fossil prep, and they were doing some prep work on a triceratops skull. And so one of my nieces started to reach through the glass, and I had to pull her back. I was like, you can't touch anything in here because every fossil is unique, and if you damage it, that's it. It's done forever. You can't undamage it. And so if there was something that we were going to learn from the section that you damaged, we'll never learn it again. And so, yeah, the ability to undestructively examine these things. Sorry about that. Forgot to push my cough button. But it's really cool that we can do that, and we've gotten a lot of really cool stuff. I've seen some of the outcomes, and also sometimes things like that end up getting put up publicly as 3D models that you can just grab and take a look at the fossil yourself.
Yeah, exactly. So the Whitmer lab, Larry Whitmer from Ohio, he does a lot of these scans. I'm doing a lot of my own, but I've referenced his work quite a bit. But they do scans of like the brain case and the cranial nerves, which can be informative of, what did a dinosaur brain look like? How big was the region they were using for smelling versus how big was the region they were using for sight, etc. So, basically inferences about soft tissue that you wouldn't expect to get from bones. There's also been a lot of work on the sinuses of the skull. So some mammals do this. Well, not some. Mammals do this, and archosaurs do it, and some lepidosaurs do it kind of in their own way. But it's essentially a sinus is a space in the skull. It has its own epithelium, it has its own tissue, but in fossils you just get the space. So, for example, like a bird has a incredibly pneumatic skull, which means that the bone has been invaded by these outpocketings of either the sinuses of the face or of the middle ear sinuses. And so the ones related to the ear are probably related to hearing, but then there are also the ones in the front of the skull that are believed to be related to cooling the skull, because, dinosaurs are pretty well supported to be at least mesothermic, if not endothermic. Many of the later theropods are endotherms. They're active, warm-blooded animals. Warm-blooded being a kind of broad and sometimes not particularly useful term. We would call a platypus warm-blooded, but its actual metabolism is very low relative to the rest of mammals, which we kind of expect, as it is a monotreme, which are not part of the main mammal group.
Yeah, the outgroup when it comes to mammals.
Yes, exactly. But reconstructing the size and shape and extent of these sinuses is particularly interesting in studying the thermal dynamics of these animals. How are they handling excess heat? Is it related to respiration and stuff like that? And the CT scan is also, there's an outstanding question whether dinosaurs have what we call respiratory turbinates, which are these very paper-thin scrolling bones that are in the noses of birds and mammals, and they're important in moisture recapture, and not all, but most warm-blooded organisms have them. They're lost in some groups.
Weren't they suspected to have been found recently in an ankylosaur?
Sort of. So there are two kinds of there are nasal turbinates, and then so there's the olfactory turbinates and then the respiratory turbinates. And reptiles have olfactory turbinates, which are spirally, just kind of like the respiratory ones. The issue is whether the elements are ossified or not. So in mammals and birds, these structures are bony, except very fragile. So in a fossil, they're almost never preserved. But what we're hoping to do, and what some people have proposed, I think there was a proposal that a pachycephalosaur, one of these dome-headed, head-butting dinosaurs that you might be familiar with, I believe it was Stegosaurus, has a ridge that would on the inside of its nose for the attachment of these turbinates, which is again something you could really only see if you CT scanned it or sliced the skull in half. And I think there is one good Stegosaurus skull, so you wouldn't want to do that.
No,
so again,
please do not.
CT scans are very helpful. Now, there is a complete side tangent in regards to these kinds of scans, but and destructive sampling, in which these scans, being kind of a public resource, are quite important. In that there's a location, I believe it's in England, I could be wrong, from the Silurian. So 420 million years ago, there were no reptiles or amphibians. Fish had jaws, and that was about it. There were maybe tiny plants and some arthropods on land. But there's this site that was, I think, believed to be an ash site, a coastal ash site. And it has these bizarre little segmented bugs and crustaceans. And what they've done is they basically sand down the fossils a bit and then get the outline and then keep sanding them down. And what they're doing is kind of similar to CT scans, but instead of scanning it, they're sanding a bit down, looking at the cross-section, computing the cross-section, sanding it down, because they can't scan it traditionally.
Oh,
so what they're doing
just manually scanning it.
What they've done is they've completely destroyed the actual fossil.
Right.
However, online now there is a 3D reconstruction of it in space, so that becomes the reference specimen, which is something kind of fascinating.
Digital reference specimen with no actual original left.
Yeah, exactly. So
the traditionalist part of me says, I don't know if I like that, but the part of me that likes high-tech stuff thinks that that's cool. So
yeah,
because
it's they're in these concretions, so it becomes kind of a trick of how do we get information out of these? Can we not CT scan them? How would we do this? I think the silicates there and make it hard, but it's been a while since I have read that.
Yeah.
But anyway, so CT scans are cool. You can look at more than you can look at brain cases. You look at sinuses. You can also look at the posterior elements of the skeleton. Sometimes, if something's not completely prepped out, there can be more interesting material in the rock that still surrounds part of the fossil that you didn't expect to find. For example, I'm currently working on a certain, I can't say which one, a dromaeosaur of so a raptor-like dinosaur, and we don't know. So we haven't found scleral ossicles in a lot of these, and scleral ossicles are the little bones in the eye, and they kind of make a ring around the. They make a ring in the orbit, and when preservation is really good, you get one of these rings. And so it's really hard to tell when these structures aren't present, because they might just have fallen apart. So we can't say it's like, oh, these things did or did not have rings. But in CT scanning one of these specimens, we found that the ring had collapsed and there were all the plates of the scleral rings still inside of the skull, which has allowed us to extract them and kind of reconstruct the ring.
Oh, awesome. Which also helps you to know things like how big was the eye, because, it's sitting in that orbit, but it's going to be much smaller than the orbit in virtually every case. But how much smaller is like one of those, I don't know. But having the ring says, okay, it was about this big.
Yep.
So that's
absolutely. And so for the sinus reconstruction, it's really and stuff like that. It's good when the sinus is completely surrounded by bone, because then you can tell the extent. But like the eye, sometimes they might have soft tissue borders, so like the suborbital sinus does. In which case, a little guesswork is involved in reconstruction.
Well, I feel like that's always going to be the case until we invent time travel.
Well, yeah. So the lacrimal sinus is entirely enclosed in the bone. So it's nice because you can get the full shape of it, or like the frontal sinus. But yeah, some of that stuff is walled by soft material. Then, so CT stuff is really cool. A lot of cool stuff going on with CT stuff. Besides that, in realms that I don't work as closely with, but that are still very interesting, is the subfield of taphonomy. And so taphonomy, subfield isn't even fair. It's its own field. It's a big field. And taphonomy is basically the study of how things get preserved. So there's been a lot of interest in taphonomy, especially with, I think back in 2004 or 5, Mary Schweitzer published her, at the time, controversial piece on preserved proteins in, well, proteinaceous structures in a T-Rex femur. And there had been reports of such features before, were met with a lot of skepticism, and in the ensuing over a decade since that has come out, there's been a ton of work. I'll plug someone I know. So someone in my who works a little bit in my lab in Derek Briggs' lab at Yale, Yasmina is like the best in the game at this stuff, and she's done some really incredible work looking at these originally soft tissues that get preserved in bone. And she's done a lot of work looking at like how proteins mature, a lot of chemistry. It's so interesting that I had to relearn some chemistry for it, because chemistry was never my favorite. But these examples of exceptional preservation are really fascinating. So for example, something that I think is pretty cool is that a lot of the times when you see these beautiful slab specimens where it's like you can see the outline of the body and like what looks like it could have been skin and stuff like that, is that it's not in the case of the T-Rex bone stuff. That is a matured protein. It is part of the original animal. But in other cases where you have these beautiful specimens, what we've learned about the preservation using high-powered microscopes to look at the texture and the structure of these fossils is that what you're seeing isn't the tissue that's fossilized. What's happened, and this is called autogenic, it involves a process called autogenic mineralization, but essentially what's happened is something dies, settles to the bottom of almost always a body of water, if you want to get nice soft tissue. And early on in decay, essentially you get microbial mats that form over it. And these microbes kind of make a mat that shapes the soft structure. And the action of those microbes, basically they're respiring and consuming of this tissue, leads to processes conducive to the formation of minerals, often phosphate. And then you get basically phosphatization of these soft structures, or sometimes pyrites involved or other things. But essentially you're getting the fossilization of these microbe mats that are the shapes of the soft structure you saw before. So very cool stuff.
Oh wow, that's interesting.
Very, very smart people figured this out. Derek Briggs at Yale, he's an invertebrate paleontologist and a taphonomist, and he's done a lot of these experiments where he just decays things to see what happens, like shrimps. He'll play with mineral concentrations and conditions to see basically how are these exceptional fossils forming, and he's very incredibly intelligent. I'm working on a little project with him right now on sea scorpions, which is a little outside of my realm of expertise, but Yale kind of likes.
That seems a little early and a little bit invertebrate for you.
Yeah, Yale likes their students to do basically something outside their wheelhouse before they become candidates, which is fine by me. Sea scorpions are very cool. So the early part of the treatment is actually in review right now for a little paper, which will be fun, and I'm sure will interest some people. It's on big eurypterids. People like big, scary sea scorpions.
Well, of course. I mean, there are like 2-meter long eurypterids out there. It's crazy.
Yes.
Or is it 1.5 meters? Yeah. It's somewhere around there.
Oh yeah, no. So Jacolopteryx from Scotland is like over 2 meters, a little. It's very big.
Yeah, it's just
there are tons of huge arthropods like these. So yeah, taphonomy is really cool. Anatomy work with CT is very cool. There are a lot of things that are very cool.
And
my
eurypterids are really cool.
Eurypterids are very cool, and you can find them if you're in the U.S. A lot of places. Now another pet interest of mine are what I call the evolutionary trendsetters that are crocodiles. And I'm going to ignore that there probably were therapsid mammals that were doing similar things before crocodiles and dinosaurs took over, after those mammals died in a big extinction. But I like this point because it's kind of a fun teaching point where a lot of the morphology and anatomy and roles that we kind of traditionally think of dinosaurs as filling in the Jurassic and the Cretaceous are first occupied by crocodile relatives that don't do super hot in the Triassic-Jurassic extinction. Some of them make it, but these early crocs are doing a lot of the same things. And then what's really cool is that later, when dinosaurs are still around, some of them start doing these things again. So for example, when we think of armored dinosaurs, we often think of, Ankylosaurus with a big club tail, nodosaurs, coelacanthines, stuff like that. However, in the Triassic, there is a group called the Aedosaurs, which are quadrupedal armored crocodiles that ate plants and maybe were omnivores, but seemed to eat plants, with armor plates and spines that were low to the ground. They weren't as big. I think the biggest one only got about 4 meters long. But, doing a very similar thing to Ankylosaurs.
And having pig snouts.
And exactly, very cute pig snouts. And then later, in the Cretaceous, a group called the Nodosuchians, which were much more closely related to living crocodiles than things like Aedosaurs were, they start doing this again in the southern, in Gondwana, so the southern hemisphere. Some of them are like that big, some are a little bigger, but they again develop kind of heavy dermal armor, low profiles, rounded faces. And it's not just, armored things. There are croc relatives that were basically filling the roles of big meat-eating dinosaurs before they were around. So things like Postosuchus and Prestosuchus were at least sometimes bipedal, probably. Also possibly quadrupedal, but they had these big, tall snouts, huge teeth, apex predators of their biome. They get replaced by theropods as major predators in the Jurassic, maybe at the very end of the Triassic. But then again, down in Gondwana, another group called Barosuchids, again still more closely related to living crocodiles than the Barosuchids, start getting big and theropod looking again, at least in their skull. And some researchers have suggested that there is a kind of niche partitioning going on where big ceratosaurs down south are the major predators, and there seems to be a lack of mid-sized dinosaur predators from Gondwana that these crocodilians were filling. I hazard caution on such assessments because I'm not completely convinced that you can accurately reconstruct niche partitioning in a lot of fossil environments unless you have like exceptional, so what we call a really good fossil deposit where tons of stuff is preserved, soft stuff, tons of fossils is a Conservat Lagerstätten. So
yeah,
a lot of the places in the southern hemisphere don't have great fossil records. We're starting to get better ones. There are exceptions like the Santana Formation, but just I hazard when you're listening to someone being like, oh, if someone says this is the niche that this thing's fulfilling, or we can reconstruct this, a little healthy skepticism is important because there's a lot of unknowns when you're doing that kind of work.
Yeah,
but again,
in order to reconstruct a paleo environment that fully, you would need to have essentially a good handle on almost everything that's living there.
Exactly.
And how can you ever know that that's what you have?
It's true. So, there are places kind of like where you might be able to do a slightly better job, like the Morrison Formation, where there's just so many fossils that you can probably reliably say that Allosaurus was probably the major meat-eating dinosaur there.
Well, there are like half of the vertebrate fossils there.
Exactly. No, yeah. So exactly. But at the same time, at the Morrison, there's a bias against smaller fossils. So it's like, do you say, oh, there aren't small predators at the Morrison, which we know is not true because we find a few of them, and they're probably not, rare in their environment.
As one of the smaller predators of the Morrison Formation. Hello.
Yes. Yeah, no, exactly. So, right, back onto crocs. The wackiest and weirdest of these, well, the two wackiest and weirdest of these kind of croc evolution tests. And I mean, there are tons I'm not going to talk about. Some of them are insectivores, some of them have blunted teeth, some of them do this and that. But the two weirdest ones are Sphenosuchians, which I am an author on a paper about them, on their inner ear evolution, which was published in PNAS, which you can check out. I provided the data for the primitive condition. But Sphenosuchians are marine crocs. Basically, the early ones look a lot like crocodiles, but with thin, long snouts. But you'd be like, oh, that's a crocodile. But the later ones, the metriorhynchoids, as we call them, have flukes on their tail. Their arms are like basically paddles. The hands have been modified into paddles, and they lose a lot of the armor on their back. So they're this fully marine group of archosaurs, and with the exclusion of birds, things like penguins, and there are a ton of birds that are pretty marine today, and hesperorniths that are extinct. But with the exclusion of birds, they're the only major radiation of marine archosaurs, because a lot of those marine reptiles are lizard relatives or sauropterygians, which we don't know exactly where they go. There's some kind of diapsid reptile is what we can
most of what I've been reading is that they are currently the most likely explanation for sauropterygians is that they are stem turtles, so they're in sort of Pantestudinata, and also that Pantestudinata goes closer to Archosauria than Lepidosauria, so I guess you could technically say they're sort of archosauriforms, which is
sort of
iffy.
Yes. Sauropterygia has been placed in pan turtles. The issue is a lot of the evidence for turtles being archosaurs is molecular. I don't often, you
they
can't.
They have thecodont teeth, sauropterygians, which helps a little bit.
Yeah, no, they do. They're
weird.
Something that often happens is that like all sauropterygians end up together in a clade. So like all these marine reptiles end up together, which might be because they have a lot of characters for marine life. So they just kind of all get sorted into this big group. We don't even know if the group is genuine. Like ichthyosaurs usually are on their own, but there's uncertainty in marine reptiles.
In case, so I did have a video about this not that long ago, but I realized sauropterygians is probably not one of those household words. So these are animals that include all the plesiosaurs and the pliosaurs, which is not a real group either, but whatever. But it also includes a lot of other earlier Triassic forms that just basically didn't make it out of there. So, including a bunch of weird things with shells that weren't turtles, because their shells were radically different. Yeah, the placodonts.
Yeah, very strange.
Yeah, so if you want more on that, check my video on turtles.
Right. Anyway, so Sphenosuchians are the major radiation of aquatic archosaurs. Depending on how marine Spinosaurus was, I guess they're not the only. It seems pretty evident that Spinosaurus is pretty aquatic. A friend of mine, Matteo Fabri, he's the best. Great guy. He's kind of one of the leading authors on that recent Spinosaur paper where they found that they had very tall tail spines. So your vertebrae have neural spines. So they're these projections that come off the top. And in Spinosaurus, on the tail, they're pretty tall and probably supported the continuation of the sail on Spinosaurus's back down most of the tail length. And they've reconstructed that as a implement for kind of a crocodile-like swimming propulsion, which is pretty cool.
Yeah.
Which would make it the most, besides birds, obviously, would be the most marine dinosaur. So most marine non-avian dinosaur.
Not quite as far as penguins, but still, it's pretty heavy marine adaptation.
Yeah, although I think it's a fluvial. I think a lot of the Spinosaurus stuff is from freshwater or like river deltas. Yeah. So they might have been freshwater. I can't remember.
We can go with aquatic rather than marine.
Yeah, I think that's probably safer. But Yeah, and so then the last group of weird crocs that, well, the last two, I'll say, group of weird crocs are these things called poposaurs, which are just bizarre. The most bizarre of this is something called Afigia, and I'm not even sure if I'm pronouncing it right. That's how weird it is. Afigia, something like that. When they first found it, they thought it was a Triassic ornithomimosaur. So to kind of describe it, it's technically a crocodile or a crocodile relative, a pseudosuchian. It has this very diagnostic ankle joint in which there's so you have two bones, the calcaneum and the astragalus. And there's a projection of the astragalus into the calcaneum, and they rotate around each other, and they let the ankle joint roll. This is not a great example, but roll, as opposed to dinosaurs, which have a hinge ankle where it can only move in two directions. Crocs can do this, and that's why they can kind of splay their hands out and they can shift their weight. Anyway, so they have this ankle, so we're like, oh, this is a croc relative. But it is bipedal, it has a beak, and it has no teeth, and it has short arms.
Yep.
It looks like a dinosaur. It looks a lot like a dinosaur. And the first one found, Shuvosaurus, I believe, was also thought to be an ornithomimosaur. They found the body of Afigia, and they're like, oh, all right, no, so this is a croc. Weird animal. Nothing, to my understanding, no crocs later in the Mesozoic will do bipedal stuff. There is a Cenozoic group of crocs that have been suggested to be capable runners, but I'm not sure about bipedality. I'm sorry?
Was it the Planogranites?
Yeah.
Yeah. Planogranites.
So,
there's even some suggestion that maybe they were unguligrade crocodilians, which would also be unique. Because
yes,
they have
that is weird. So again, even like after the dinosaurs, crocs are still doing plenty of weird stuff. But this is, in my opinion, probably just the strangest in how that's basically a dinosaur. And dinosaurs were around, but at that point, none of them were completely toothless and had a beak. They were the first to do that, and then that became something you see happen quite a bit in later dinosaurs, and it kind of begs this question of, when different evolutionary events happen, we often call it a convergence, right? They're converging.
Oh, Alex? I think we lost Alex. Uh-oh. Well, hopefully we get him back. I'll see if I can send him a message elsewhere. See if he can, Sorry, I'm just writing Alex a quick message. I think we lost him. I'm going to take him off air. Hopefully he'll come back. So yeah. Sorry about that. That's unfortunate. Hopefully he comes back. But I wanted to say hey to everybody in the chat. I know I didn't get to earlier because we were sort of jumping right in. So I hope. Oh, he's back. All right, let's get him back in.
I had to move my laptop because I think for some reason the coverage I had in there just gave out. So I'll be standing a bit. So that's fine. I can't remember what I was talking about.
You were just talking about the fact that poposaurs had converged, or we call it convergence, because they looked so much like ornithomimids.
So in the discussion of things like convergence, there's often the question of if something is deeply homologous. And what that means is basically, is it homologous? But less obviously. And what I mean by that is sometimes structures can share deep genetic or developmental controls in which, so the photoreceptors in almost all animal eyes, even though each eye kind of has a separate, not each, but major groups of animals with their eyes have different evolutionary trajectories, nautiloids versus chordate eyes, our eyes, stuff like that. Mm-hmm. But they share these photoreceptors to, they share a deep homology in the photoreceptors they use. which suggests some kind of common origin. So in the case of these archosaurs, you know, I don't know if anything's been written about this, probably, but you know, it's why are archosaurs, bipedal archosaurs, losing their teeth and growing beaks so often? Because it happens in ornithischian dinosaurs, so in ancestors of things like hadrosaurs, although they don't often lose their teeth. It happens in ceratosaurs like Lemursaurus that start out as little predators with teeth, and then as they grow, they lose their teeth and get beaks, which is absolutely insane. And then it happens in a ton of silurisaurs like ornithomimosaurs, oviraptors, mononychids, although they don't really develop beaks, and then possibly therizinosaurs, which still do have teeth. But you see kind of this.
It happens over and over again.
Exactly. And that's that kind of brings me, just in thinking of deep homology, to something I'm not sure a ton of people are familiar with, which is the possible deep homology of feathers. So, as someone who is interested in theropods, I'm interested in feather evolution. I'm not the most knowledgeable about it. I do a lot of, I'm trying to keep up to date on it. It's a lot of interesting stuff. But as I'm sure a lot of people are aware, a lot of dinosaur fossils we find that are exceptionally preserved have preserved feathers.
Mm-hmm.
And some of these are like feathers we see today, kind of, you know, pennaceous or branching and stuff like that. Some are weird and unlike anything we see today in Scansoriopterids and things like that. And then some are these monofilaments, so basically single filament structures that are what we call protofeathers. And these are in the more primitive clades of theropods that have these feathers, as well as kind of dispersed throughout the dinosaur tree. They're in ornithischians, which are, so the plant eaters, if this is the tree, and then there's sauropods and theropods. And then, so you have feathers in theropods, you don't have them in sauropods that we know of. And then you have ornithischians outside that also seem to have feathers. And that's using kind of the traditional dinosaur phylogeny. But the question there becomes, are these filamentous structures deep to dinosaurs, basically? Is this something that the first dinosaur evolved, or possibly even further down, because it appears that some kind of feather-like structure is present in pterosaurs, which are not dinosaurs, things like pterodactyls and quetzalcoatlids, as pterosaurs. Quetzalcoatlid,
I think that's what's on your hoodie. Is he a pterosaur
on
there?
This is Sungaripterus.
There you go.
Which is a pterodactyloid, and it's eating a fish. But right. So pterosaurs have this structure called a pycnofiber,
mm-hmm.
which are monofilamentous mostly, and possibly branching in some pterosaurs. It's been, at first it was thought they were a separate thing, and now there's some evidence coming out that they're probably homologous to feathers, which would indicate that filamentous structures are present pretty far down. So,
at least back into Ornithodira, right?
Exactly. And this is what's kind of cool, is that someone was like, well, if it's this deep in archosaurs, it should be something that maybe the ability to form a filament is something that you can find pretty deep in Archosauria. So what someone did was they took essentially the products of genes that develop feathers in birds, these chemical signals, and what they do is they subjected crocodile, or maybe it was alligator, I think it was alligator, epidermis to it, so scales basically, in development. And what happens is when you submit gator scales to these chemical signals, they develop into monofilaments, kind of these filamentous projections of epidermis. I can't remember the authors of this paper because it's really cool and I recommend it. But
I included I did an updated version of my birds or dinosaurs thing. It actually shows some of the figures from that paper, because it's right there. It's like, look, here's an alligator growing what's basically a proto-feather.
Yeah, no, exactly. So there's a lot of deep, what we see in the fossil is the expression of, is the phenotype, right? The expression of the gene. But it's really hard to tell in a fossil, like what are the developmental controls? How is the structure being shaped? What confers this morphology? And so for that, you have to look into the genes of living organisms. But alligators and birds are pretty similar in some ways. I mean, they're very different in other ways. But they
Right.
they both have trichromatic vision. They both vocalize. Baby alligators use kind of a birdy keratin in their scales. A lot of stuff. Now I know I've talked a lot about how crocodiles are weird and cool and a lot like dinosaurs. So to do a complete 180, I'm going to now tell you that don't think of crocs as dinosaurs. And here's why. It's very common and appealing to paleontologists, sometimes professionals, sometimes, when you're thinking about this kind of stuff, to be like, all right, so birds are really, really evolved, derived dinosaurs. They're very, there's a lot of dinosaur stuff in them, but they're pretty weird.
Yeah.
What about alligators? Alligators look more like a reptile. That's true. Or at least like, what you would expect a reptile to look like, something scaly, something reptilian. But the issue with that is that they have their own super specific evolutionary path in that they are super adapted to living in freshwater. Their freshwater amphibious ambush predators is like, they're hyper adapted to that. And their ancestors are long-legged, quadrupedal, cursorial, running, greyhound-looking things. It's called sphenosuchians, which are pretty different than their ancestors. So there's something in that we do called phylogenetic bracketing. And the idea is, if a structure is present in the most recent sister group, well, the sister group is the most recent outgroup of the group you're interested in. So if dinosaurs, for example, if you're like, hey, we're dinosaurs doing this, a educated guess you could make from phylogenetic bracketing is like, are birds doing it? And are alligators doing it?
Yeah, and if they both are, then probably.
Exactly. Maybe. Probably. Theropods are easier bet than like an ornithischian just because they're closer to birds, but exactly. If only birds are doing it, you don't know. So like brooding, sitting on your eggs, for example, only birds are doing that. And we had to find fossils of oviraptors sitting on their eggs to know it's like, oh, okay, so this shows up in dinosaurs. Gators heat their eggs with decaying vegetable material. Alternatively, sometimes people will suggest that crocodile anatomy, or usually soft anatomy for dinosaurs. And the issue with that is, again, as I said, alligators and crocodiles are very well adapted for the niche they fill. And they're not dinosaurs. They are
Right.
secondarily cold-blooded. Their ancestors were pretty active. They seem to have faster growth. I wouldn't say they were maybe warm-blooded, but they were probably mesotherms. And that's something that gets lost in crocs. So you don't want to draw too many inferences from alligators and crocodiles about dinosaurs, but they can be very helpful. One of the more infamous comparisons that have been made are about their breathing. So birds have a very efficient respiratory system, which is essentially a system of air sacs, outgrowths of the lung that invade the axial skeleton, and basically let birds, when they're breathing in, they have air in their lungs, and some of that gets moved into the air sacs, and when they exhale, I'm exhaling for dramatic effect, they lose the air in their lungs, but the air in the air sacs flows into the lungs, so they're constantly keeping their lungs oxygenated in this fantastic loop. It's very impressive. They ventilate their lung with air sacs and by rocking their sternum. Crocodiles ventilate their lungs using a kind of diaphragm. So like with us, we have a muscular wall under our lung that gets pulled down.
One second, Alex.
But
We're going to open up questions at about the... actually, probably after this point or so, we'll open up for questions. So,
sure thing.
yeah, because now we have a whole bunch of people asking if they can ask questions. So it is called Ask a Paleontologist. So after this one, I think we'll go to questions. So I'm sorry, finish.
Sounds good. anyway, so when you breathe, muscular wall, your diaphragmatic air gets pulled in, air gets pushed out when they go back upwards. In crocs, they have a muscle that is attached to their liver that also attaches to the lungs, and it's called a hepatic piston, and it's basically their organs are being pulled back, making room for the lungs, and that's what fills their lungs. So some authors suggested, a group that don't think that birds are dinosaurs suggested once that, based on the Sinosauropteryx fossil that appears to be misinterpreted, they had a kind of a hepatic piston because other archosaurs like crocs have it. This is not the case. Dinosaurs have invasions of their axial skeleton, just like birds, by air sacs. They have a pneumatic hiatus in a few of them, which indicates the presence of abdominal air sacs. And they have these transverse processes that would not let you pull your organs around like that unless you wanted to cut them up. So again, just to reiterate, crocs are really cool and they're doing their own thing and they're not dinosaurs. Although sometimes they can be sort of helpful to tell us about some things dinosaurs might have done.
Yeah.
I'm going to grab my drink. I'll be like a second, but I'm ready for questions.
Okay, awesome. So we're going to do questions the way that we have done these before on other shows where I have a sort of guest to interview. So get your questions in the chat. I will be going through them sort of from now on in sort of the order I get them. Although, if you super chat, that does put your question to the top of the line. Depending on how many questions we get and how long we go, I can't guarantee that every question will be answered, but I can say that I will at least make sure to ask every super chatted question. So that is the safest way, but it is by no means necessary. And so I'll actually start with this question that I got from Vandelli1998.
Hello.
Hello. So let me just make sure I pull up the little window, so in case the super chat does come through, I can make sure I get it even if I'm not scrolled to that point. Okay. So this is a question for Alex from Vandelli1998. There seems to be a variety of this is brants in the past. I'm not sure what of crocs in the past. How come there doesn't seem to be a variety like that anymore? Yeah. So why is it that crocodilian or even just pseudosuchian diversity seems so low right now?
That's a good question. I would say it doesn't even seem low. It is pretty low relative to the past. Why? Well, like any group, there are ups and downs. And maybe Sarcopteryx, so lobe-finned fish would be a good example, but for crocs, they are the group that takes over after the Permian extinction. Right? So they kind of run the show for a while. Then there's a big extinction at the end of the Triassic that really hits them. Only a small group makes it through, the crocodilomorphs. They continue to persist. Then they're in the shadow of dinosaurs, so it's harder to do as much because there are dinosaurs around. But they still do pretty well, and throughout the time of the dinosaurs, they really start expanding into all these interesting niches. Some are marine, like we said before. Some in the southern hemisphere do a lot of crazy stuff. Then there's a big rock from outer space that slams into the Earth. And from what we can tell from looking at, what kind of animals end up surviving the extinction at the end of the age of the dinosaurs, it's that it's usually mid-sized to small freshwater organisms. So turtles do pretty good. Mm-hmm. And it seems that those crocs that were living in freshwater realms were the ones that had the best chance, or not best chance, but survived that extinction event better. That could be a bias in the fossil record, but based on what we see today, seems likely. Then, after the dinosaurs go extinct in the Cenozoic, there's a little bit of kind of this interesting diversity that happens early on when it's pretty warm, but then we get a glacial event, and we just kind of see these amphibious ambush predators persist till then. So they've taken a few beatings and licks in their time. And now today it's mostly semi-aquatic ambush predators, but some, like gavials, are very specially adapted for fish, and some caimans are more terrestrial than alligators. But I guess the simple answer would be there have been a bunch of extinctions and climate changes that were not favorable to the more weird, specialized terrestrial crocs.
Okay. Let's see. This is from Andrew Cumming. My question for Alex: what is the importance of transitional forms? Oh, 1 second. Just had to scroll. What is the importance of transitional forms if they are mostly side branches, not direct ancestors? Does it have to do with acquisition of traits and such?
That's a good question. So, in phylogenetics, which is basically how we produce our modern taxonomy, the production of, you've seen evolutionary trees, except in some cases with like plants or immediately observable speciation events, phylogenies show relationships and not ancestry. And that's important because everything is a side branch, so to speak. But in your question, you kind of allude to the answer, which is that what a tree really breaks down to are character trait distributions. So distributions of the character states at every node, which are the internal points where the branches meet, and at the end. So it is entirely possible that you have a fossil taxa that is from the right age, and let's say it is literally an ancestral organism to something up on the tree. It would still come out as a side branch just because of how the trees are constructed.
Yeah.
However, what you could do to kind of check, or is like, how similar is this to the ancestral state, is if you have a really well sampled tree with a lot of morphology, you have the character states of the things on your end, so thousands of characters they're scored for. But then you also, where they meet their nodes, at least in parsimony, those are averages of those states. So it's like the ones where they're all the same are going to be the same, and then there are going to be differences basing on how you polarize your tree. But what you can do is you can do this ancestral state reconstruction, where you're like, okay, the common ancestor of this and this, based on the characters in our tree, should have these characters, these morphological traits. And what you can do is you can look at your tree and you're like, oh wow, so this sphenosuchian is almost identical to this node. That organism is very similar to what we would expect the hypothetical common ancestor to the next branching pair to be like.
Right.
It's when we're talking about transitional fossils and intermediaries, it's really a discussion of characters and less so the organism itself. We're interested in, how are these character states evolving over the tree? I hope that is helpful.
All right, and for $5 from Java Man, we have, in the field, how do you tell a dinosaur bone from a rock? And when are you removing? And when you are removing the fossil from the rock, how do you tell the two apart?
Good questions. So depending on where you are, the most time. So I've done field work in South Africa, Lesotho, and in the Yunnan province of China. And in some places, like in Yunnan, there are fossils all over the place. A lot of them are pretty scrappy, but they're all over. In other places, like in Lesotho, you can go for a while without seeing something, but then when you find something, they're all over the place. And then where we were in South Africa was pretty spotty, but it was a new site we were checking out. There's a ton of great fossil places in South Africa. But simply, bone is usually a different color than the rock it's in. It will so for example, in South Africa, there's a lot of this red stone, these kind of paleosols, and the bone is white and often kind of purpley and red in coloration. But a kind of a classic paleontology story is that if you're really not sure, you can lick it, because bone, although it is fossils, although they're replaced by mineral, will still preserve the little pores in the bone. So if you lick it, it'll stick to your tongue a bit, and a rock won't. They're slightly radioactive, so fossils are sometimes slightly radioactive. So people are like, oh, be careful. But I mean, as long as you're not eating them, like by the fistfuls, you'll be fine.
Yes, dude, that is the last thing you want to do with a radio source is eat it.
So yeah, so the first trick is like, am I looking at, is this weird thing I found, this tiny flake or this weird misshapen thing, is this bone or rock? If you figure out it's bone, and it's usually disarticulated and kind of on the surface, you're like, okay, is this just something that's wet? What is this? And it's usually that there is more bone somewhere else that weathered out of, that you found kind of on the ground. So you try to follow the flakes. Find the flakes, you follow the flakes, and hopefully when you follow the flakes up, you'll find where they weathered out of, which is usually more of them. And it might be an articulated skeleton. Now, once you find the fossil, you need to know the anatomy of the organism. So, for example, we recently excavated a kind of croc from one of those sites we were in. And if you didn't know better, because I mean, I worked on crocs, and this was earlier in my studies. Like, I could see the skull. Like, the skull is like, oh my god, that's the skull. We had cleared away a lot of the rock on top of it. The tip of its snout was pointing out. But then there were all these weird things on the top where I'm like, what are those? And it turned out to be their osteoderms, the armor on the back of the croc.
Oh, okay.
And, once you've done that, you figured out where the skull is, and you figure out the direction the skull is pointing in. And then you're like, okay, so the rest of the animal, if the skull's that way, the rest of the animal's probably that way. And then the arms are somewhere. So what you want to do is you basically have to slowly chip away at the outsides and the top very carefully, because you want to see how far the arms and legs go and how far back the rest of the animal goes, because you don't want to lose. You don't want to tear off or break anything.
Right.
And again, like, if you sent me to an invertebrate site where there were soft fossils, I'm sure I'd lose some incredibly important worm or something, because it's something where you really need to know what you're looking for, or else you run the risk of breaking stuff.
Yeah.
And that happens even with professionals. One of my advisors says, his least favorite part of field work is getting the fossil out, because you have only yourself to blame for breaking stuff.
Yep.
So be very careful.
Now, this reminds me, speaking of fieldwork, are you familiar with a paper by one Mark Armitage on a supposed Triceratops horn?
Yeah. I haven't read it in some time. I read it kind of a while back when it was making a secondary rounds. Didn't he have another paper that came out recently?
He might have. But
I think he's got more supposed Triceratops stuff, yeah.
So, in the original paper that he published on this subject, which was in a photography journal, I basically roasted him for garbage methodology, but I'm not a paleontologist. I want to know, was I right to completely lambast his apparently terrible methodology where there was no site map, there was a reference to a formation that doesn't seem to be in the literature, there was only one in situ photo taken of one of the three specimens that he claims to have found, there was no analysis to determine that it was a Triceratops evident in anything that he wrote? Is that?
Yeah, that's not great. It's not. So, for example, we have a paper that's we're working on right now that is a monograph of the anatomy of one of these early crocodiles. And the first part of it is I didn't even collect this thing. This thing's been in a collection since 2006. But the first part of the paper is, where's it from? What are the GPS coordinates that we dug it out of?
Mm-hmm.
What horizon in the formation is it in? What's the date based on this, whatever ages have been worked out for the formation it's from? What's, and then pictures. We don't have a ton of pictures in situ. We do have pictures of the entire animal. And then the rest of it is like, here's the evidence that this is what we say it is. Like that. It's not good methodology not to have, you want everything to be detailed because you want someone to be able to check your work.
You mean you didn't just grab a bone and take it home from somewhere and then toss it in a bath of acid to see what happens? Because that seems to have been his methodology.
It's not great. No, that's not great.
Yeah, I just wanted to... So, this has been a little bit of a theme for a little while because some creationists that I have interacted with like to bring up Mark Armitage finding all these sheets of soft tissue inside of a horn. And basically, the conclusion that I've come to is this is probably a bison horn.
I've heard that.
Yeah.
That would be incredibly funny if it was.
Well, so there is actually a species of bison, bison latifrons, which can be found, obviously not in the same geological setting, but geographically in the same types of places. So basically, you go uphill a little bit, and you'll find bison latifron skulls, and bison latifron horns are actually very similar in size and somewhat in overall shape to Triceratops horns. So if you find an isolated horn, you have to be pretty careful, and there's no indication that he was. And so we also can't check where he got the horn. And I mean, frankly, it's a young Earth creationist, so you expect them to get basically everything wrong. Because you can't be a young Earth creationist without being wrong about almost everything in science.
I'm almost sure he had a second paper come out recently.
I'll have to check into that. But
in my crocs journal.
We have something from Nestle20 who says, I have seen one paper that proposes that heterodontosaurids are stem pachycephalosaurs, and another paper suggests silosaurids are basal ornithischians. Do you know anything about that? Yeah, so that paper is pretty recent. The proposal that silosaurs are the missing Triassic ornithischians has been around for a little while, and heterodontosaurs. Well, heterodontosaurs are something. They are definitely something.
Yeah, they're probably one of the most primitive groups of ornithischians, but some have proposed that they are the deep missing relative of Marginocephalia in the Jurassic. Well, for pachycephalosaurs. I am not an ornithischian person. I read that paper. Read a lot of that paper. I haven't read the whole thing because there's quite a bit in its supplement. I can tell you the following. Ornithischian phylogeny is currently a really hot topic. And
it's a mess.
Yeah, so for comparison, theropod dinosaur phylogeny is pretty good. It's been kind of this huge area of study for like 2 decades, and there are still questions. But like the shape of the theropod tree is pretty good. Ornithischians and sauropods, however, they're not bad, but there are some questions that remain in interrelationships of some groups that can change the tree around. Heterodontosaurs have been a bunch of things. I've seen they've been proposed to be kind of the sister group to our basically horned and dome-headed dinosaurs before. They usually end up outside of it. It's a pretty substantial analysis. It's kind of the first of these really huge ornithischian analyses. So I'm sure they certainly got plenty right. I don't know enough about the characters that put heterodontosaurs in Marginocephalia. I would recommend caution, and we should see what further work using their matrix produces with more taxa scored and stuff like that. It's possible.
I tend to lean towards heterodontosaurs being basal ornithopods, and that's just sort of based on the overall. But again, I'm not willing to take a stand on that. I'm not going to fight anyone who says that they're not basal ornithopods.
Yeah, so they often get treated as primitive ornithischians, and they're often found as primitive ornithischians, which are 2 different things. They're pretty weird and derived in their own right, so you don't want to assume that's what the basal ornithischian looked like. Which brings me to silosaurs, which if you don't know are kind of these weird herbivorous dinosaur relatives, or maybe apparently ornithischians that are in the Triassic. And one of the issues is we have sauropods and theropods from the Triassic, but we don't have ornithischians. Well, maybe. Depends on what a few things are.
We seem a lot of
the things that seem to be ornithischians early on were then reclassified as silosaurs. And a little bit by going back and forth over the last few years. They have a lot of the features that put them as ornithischians are in their teeth. And I am always a little skeptical of anything that's not mammal. So mammals have really great dental fossil records. But in reptiles, there seems to be a lot of variation, and I'm not totally sure if those tooth characters are super strong. But again, they're so close to the base of dinosaurs, it's entirely possible. It's what we call a kind of a zone of error. I like to call it the great evolutionary smear, where once you get down to like, and it's a great example of evolution, it's basically once you're getting to the most primitive members of all these groups that are like the first of these things, the issue you're going to have is they're all going to look really similar.
Yeah. Actually,
they're pretty general.
my friend Jackson Wheat, who has another YouTube channel, and he does a lot of biology and evolution stuff. I inspired one of his more recent videos where he's taking a look at, you can take these organisms that today don't look very much alike. Like, so you take, say, rhinoceroses and horses. No one's going to confuse these two animals. They look extremely different. They act extremely different. They're just not very similar animals. But then you say, okay, what about the first rhinos and horses that we get in the fossil record? Oh, they're basically identical. Or you look at, say, elephants and manatees, and you say, well, they're extremely different. But you look at the first Sirenians and the first Proboscideans that we have in the fossil record, and they're basically identical. And so, it's a really cool thing. The further back you go, the closer these, what you now think of as very different branches, the more they look like each other.
Exactly. And that is kind of what we should expect. It'll be hard. It's hard to kind of figure out what a lot of these basal things are. So Eoraptor is pretty famous, and when they first found Eoraptor, everyone was like, oh, it's a theropod. And now it seems like it's a really, really primitive sauropod. That's like basically, if you were curious what the earliest dinosaur looked like, Eoraptor is pretty close to it.
Yeah. Let's see. This is also from Leslie 20. The teeth at birth and the beak at adolescence, is that a type of ontogenetic niche partitioning? Maybe? Meaning they had different diets at different stages of their life?
That's what's proposed, and I think where sometimes, niche partitioning arguments, there are weaker and stronger ones. I think in the case of Lemosaurus, that's a pretty good one, because
it's hard to figure out what else that would be the result of.
Yeah, no, exactly. It's actually incredibly rare to see that degree of kind of feeding strategy change in a tetrapod. In vertebrates, I think there are some fish that do it, and maybe a few amphibians. I could be wrong, but it's crazy that these animals that are the juveniles, which are these kind of little ceratosaurs, big teeth, munch, probably bugs. They lose them as an adult, and they develop beaks as adults, and that they're eating plants. I actually, when I was at GW, Joseph Stiegler, that was he was working on all this Limosaurus stuff, and I've played around with some of those skulls as far as CT segmenting, and they are really weird, but pretty cool.
All right.
But yeah, probably the adults were eating something different than their young.
Okay, so let's see. I'm gonna scroll down and see what other. So this is again from Nestle 20. This is mere speculation, but could Spinosaurus use its body, back and tail sail as sort of a dam to redirect fish to its mouth? Maybe that's what the sail is for? I doubt that it could do that. It seems like an extreme range of motion that it probably didn't have, but I'm not an expert on Spinosaurus. What do you think, Alex?
No, neither am I. But I think that would probably enter into the realm of speculative biology.
I would be pretty surprised. I think one of the
so one of the issues is theropods have accessory articulations in between their vertebrae. They're called hyposphene and hypantrum articulations, and they make a really rigid axial skeleton. And one of the cool pieces of evidence that supports the Spinosaur tail might have been for swimming, for lateral motion, is that it lacks these secondary articulations. But I don't know if the rest of the skeleton does. So,
yeah, I think one of the things is that I find humans have a pretty strong bias towards thinking that other things can move like they do, but humans are actually absurdly flexible compared to most animals. And mammals in general are also more flexible than dinosaurs were in most ways. The big exception being the neck. Dinosaurs had pretty flexible necks that are more flexible than most mammal necks. But other than that, as a general rule of thumb, if a dinosaur is going to be less flexible than a similarly sized mammal.
Yes.
So be careful when you're thinking about like, oh, if a tiger could do this, then maybe this dromaeosaur could. It's a fair chance no.
Exactly.
Yeah.
Also, I'm moving again because I need to plug in my laptop.
No worries.
The Wi-Fi can be fine where I'm moving.
You might think that this is a big disadvantage, being less flexible, because you have a lower range of motion. And while that's true, it's also important to note that lower flexibility all comes with the benefit of being less prone to injury. Like, dinosaurs probably didn't sprain their ankles as much as humans do, simply because their ankles could not move that way. And so you don't get that injury. So there is that sort of trade-off between proneness to injury and flexibility. The more flexible you are, the more likely you are to be injured. So, there's that give and take. And mammals have gone for the more flexible but more injuries, and dinosaurs seem to take the less flexible but fewer injuries kind of dynamic. So let's see. Sorry, there was just a bunch of things that were not questions, and now I'm just scrolling down so I make sure I get the actual questions.
No problem.
Okay, just a quick follow-up. This is from Andrew Cumming. If it's acquisition of traits, this is reference to transitional fossils.
Sure thing.
What does it mean that they link two or more groups? And I think he's asking, like, when we say that this is say, Tiktaalik is the link between Sarcopterygians and later tetrapods.
So trees are what we call nested hierarchies, which are you have a broad group, and then they get smaller and smaller and smaller as you move up the clade. Although, smaller doesn't always mean there's less stuff in it. But, tons of beetles.
Mm-hmm.
I guess actually there would always be more things in the clade outside. Never mind. Yeah, so clades inside will, by definition, have less stuff than the clade they're a part of, because the clade they're a part of contains them and the other stuff.
Yeah.
My bad. But yeah, basically, so in the case of Tiktaalik, what becomes important are what we call shared derived traits, or synapomorphies. Essentially, features that tie these things together. So in Tiktaalik's case, Tiktaalik is a stegocephalian fish. Technically, it's not a tetrapod. And it has a lot of primitive features, and primitive features don't provide grouping information. For Tiktaalik, it's all relative. If you were talking about like a more primitive lobefin fish, what you consider primitive in Tiktaalik might be important. But in Tiktaalik's case, relative to the rest of its features, it has primitive features like large, heavy scales. It doesn't have a true neck. It lacks an occipital condyle. It doesn't have digits. It's got ray fins. So these don't provide specific grouping information for where Tiktaalik goes, because it's in all this other ancestral stuff. It lets us know that it's part of these ancestral group, this much bigger clade. But for specifically where it goes, we have to rely on shared derived features. So specific things it shares with other derived clades relative to itself. So in this case, wrist bones, choanae in the palate, these are things that, relative to those plesiomorphic traits, those primitive traits, are derived. And based on the derived traits, we can group things together. So when we say that, tetrapods are, Tiktaalik is a kind of tetrapod, or it's a choanate fish. We'll start with that, in that it is a Sarcopterygian fish that has choanae. And so it's part of this clade that is defined by the presence of that structure. We would call that an apoclade, an apoclade. This also includes more primitive things like Eusthenopteron. And then as you move up, there's things with a head separate from the neck. And that's, again, so now Tiktaalik's even closer to tetrapods. So it is close to tetrapods, but it's not a tetrapod. And that depends on the presence of these shared derived characters. I wish I had something to draw with because that would help me explain it better. So yeah, the key is not commonality. The key is derived specific characters that occur with the congruence of the rest of the tree. Those primitive traits are not unimportant. The congruence between Tiktaalik stuff and those other fishes stuff is shaping a lot of the tree. But for where Tiktaalik is specifically relative to more derived stuff, that shared derived trait is very important. And that applies for literally every organism anywhere in a phylogeny ever. Even with molecular evidence, instead of features, you're doing it for nucleotides. So it's like, what's the important shared mutation in the mitochondrial DNA here that links these together? And it doesn't always have to be adaptational either. Sometimes it can just kind of be.
Right. Actually, a lot of genetic analysis tends to focus heavily on stuff that isn't basically neutral variation, because non-neutral variation is under selection pressure, and so you can't make as many predictions about exactly whether or not that genetic similarity is going to be because of selection pressure or recent ancestry. Whereas with neutral evolution, where you just get things like synonymous codons or non-coding regions that are basically just buffers for genes, changes in there are much more likely to simply be the result of random mutation and inheritance of those random mutations without any selection. So that's just a little quick thing from the genetic side there. But let's see. Okay, I don't think that was a question for you. Let's see. Okay, so this is from Nestle20. I have seen one paper that... Oh, that was already read. Sorry.
No problem.
We had some repeats because... Okay. Nestle20 says, that comment about at the base they all look very similar. I send an image on Discord to you. Please show it. I think it will show how different dinosaurs are all closely related. So give me a second. I will pull up my Discord, and we will take a look at that. Sorry, my Discord is closed so as to not give me weird noises while I am streaming. So, you guys didn't think I was going to get to your questions, but I did the first time. Okay, so let's see. Okay, here we go. Let's... I'm going to try and pull up the full resolution version. There we go, open original. Okay, so let's share that. We can take a look. Give me a second. So... we'll go here, and we can pop that up there. And so, yeah, so we're starting up at the top here, where some of the most primitive forms in Dinosauria, and we're showing how they are essentially just the same thing. I mean, come on, these are very simple. Yes. Yeah, like these three organisms, you would. They're the kinds of things a creationist might say they look like they're the same kind of animal. But then as we get down, because going down is going forward through time, we're getting like, okay, so very first sort of sauropodomorph type things. Oh, we're getting more sauropody, and then suddenly we're down here, and it's like, oh, well, that's very different. And then we're getting, you know, the same sort of thing in like therizinosaurs. Tyrannosaurs,
that's a good one. There's I'd stick Alvarezsaurs in there. They look very nice.
Yeah. And so we're getting the same thing with ornithopods, where we're getting. I think this isn't Hypsilophodon a heterodontosaurid. I think they're going with my.
They're probably... No, Hypsilophodon is a ornithopod of uncertain, a neornithian of uncertain placement.
There you go. But then so ornithopods and Ceratopsia, and you're getting the same. We're diverging as we go.
Yeah, Ceratopsia's got a great...
We're getting Pachycephalosaurids and Thyreophorans.
Got a nice one too.
Thyreophorans are animals like Stegosaurus and Ankylosaurus. So yeah, see, and then we get the split, right? So up here over at Scelidosaurus, we're still sort of in the basal area of Thyreophora, but then you go one way and you get towards Stegosaurus. You go the other way and you get towards Ankylosaurus. And so
Right.
yeah, thank you very much, Nestle 20. This is a great little illustration because it really does show you what it means for all of these to be very closely related. But then you get down here to like the end Cretaceous, or in the case of, you know, Stegosaurus, the Jurassic. And like, these are all very different animals. Wasn't always the case. So all right, I'm gonna take that screen off. So yeah, that was a really good picture. Thank you very much. That was very good. Okay, so this is from Andrew Cumming. Just a quick follow-up, a second one. Oh no, this is the same question. So, guys, I got your questions. Come on, guys. They have no faith in me. Let's see. So last thing, this is from Andrew Cumming. So Alex is saying that the transitional fossils share traits with some groups and some with others, and that reveals relationships through data analysis rather than ancestry.
Yes.
Yeah.
Yeah. So transitions and anything like that, we often refer to as mosaic character trait evolution, which is some things change before other things. Things seem to change at different speeds, which, you know, would make sense.
Mm-hmm.
But that all has to be inferred from a tree topology. So before we can say anything about you know, what's related to what and how this character evolved, it has to be interpreted in a tree. We need a layout, and that's how we make inferences about it. Which is also a nice little thing to remind. There are some individuals who are critical of evolution, who are like, you guys are just lining up fossils, which we are not doing.
No.
I have to code 400 animals for 1,200 anatomical features, and then I get a tree, and then I have to run that analysis like 24 times to test all the parameters and see what groups we get, and then I can start saying things about relationships.
You're not just eyeballing a bunch of fossils and then writing down whatever evolution you think happened?
Correct. I'm not doing that.
Yeah, it's almost like this is a science with methods and, you know, lots of mathematical rigor. Almost.
Exactly.
Yeah. So we have a question from Vandalia 1998. Says,
Sure.
do we know why the pterosaur branch got to the air before the dino branch did?
I mean, do we know? Like, I say probably not. My suspicion, though, and I think this is probably pretty reasonable, is that they were just first. Because they're one of the more primitive not primitive, they're one of the earlier branches of ornithodyrans. It goes, so we have crocs on the side, and then we have sphenosaurs, which are quadrupeds, and then most of the time we get pterosaurs next, and then a bunch of dinosauromorphs and silosaurs.
Yeah.
So, I mean, it could have been the availability of an aerial niche. Again, I don't love those kind of adaptation niche explanations, but it's possible.
Well,
there were only birds. I'm sorry, not birds. There were only bugs flying around. So, you know, it could have easily been like, hey, there are bugs flying around. This isn't too bad. It would be very nice to know what the intermediary pterosaurs look like. That's one of the current hot topics for research. Is the earliest pterosaurs are very pterosaury, eudimorphodon, stuff like that.
Yeah, they start out with a big set of the suite of adaptations that you're going to find throughout the whole of Pterosauria.
Yeah, exactly. And once you get into pterosaurs, there are really cool transitions. Like, you can see the transition from short-snouted, tailed things to like what we call monophenostratans, which big heads, long necks, they lose the tail. There are good transitions within pterosaurs. There aren't good we still don't have a nice, it's kind of like bats. We're missing this early transition, and it happened quickly. The animals that were transitioning lived in a different environment. like a mountain. You don't get a lot of fossils from a mountain because it's eroding sediment and not often depositing sediment, or a deep jungle that fossils aren't often well preserved unless you have a nice lake. So there are reasons we don't get. We're not totally sure. But my suspicion is that the first flying pterosaurs are probably a little bit older than the first dinosaurs. But it was all happening pretty quick in the middle Triassic. So
yeah. Well,
maybe
one thing I would like to say is that the question comes up, I think, is partly sort of an artificial factor of how we name organisms. Because you gotta remember, the fact that Tyrannosaurus and, say, Triceratops are dinosaurs and pterosaurs aren't is basically because Richard Owen said so.
Yeah.
So you could just say that they're on the same branch. They're both ornithodyrans, and it's just that pterosaurs are the first ornithodyrans to fly, and birds are the second one.
Yeah, that's a very reasonable way, and I'd also put that. So we have flying pterosaurs in the Triassic, right? Mm-hmm. Plenty of them. Well, I mean, a few Preondactylus, stuff like that, and then we have, as opposed to birds and pterosaurs, we have a good series for birds, for the arboreal flying. That transition is pretty good. It's one of the best. In my
at this point. It's been getting nothing but better lately.
Exactly. But one of the interesting things we see is kind of this weird smear of flight strategies that emerges in the middle Jurassic. So you have weird things like Scansoriopterids that are flying with bat wings. And then you have a bunch of weird possible gliders. Troodontids do it. If Anchiornis is a troodontid, dromaeosaurs do it. And then, of course, you have early birds. So I think there's good evidence that the middle Jurassic, probably the mid-middle Jurassic, and maybe even the early middle Jurassic is kind of where all the evolution of birds is happening, which is where we see a lot of the important fossils, which would be, again, pretty well after at least, for at least 40 million years after, probably more. Yeah, pterosaurs are what, 230? And birds are 160? So it's substantially more than 40 million years between the two. There's some arguments that birds outcompeted pterosaurs that don't seem to hold up, which again might be preservational biases.
Right.
But yeah, no, interesting question.
Okay.
I hope we find transitional pterosaurs. That would be super cool.
All right, so I'm going to make sure I'm on the right next one. Okay. This is from S. Bendit, and they ask, are there any publicly available resources to generate trees from characters? I believe the software is, you can just get it, can't you?
Yeah, so most phylogenetic softwares are free. So if you download RStudio, if you're familiar with R, I am very bad at R. I have to relearn it every time I want to use it. I'm trying to get better. R is often used for tree construction. It has a lot of packages, a phyllo tool for constructing trees. I'd have to double check the list of softwares. I use Mesquite and TNT. Both are free to download. Mesquite is a matrix tool, essentially, where you put in your taxa, and your characters, and you score them. And it's really nice because once you get a tree, you can go back and you can copy that tree into Mesquite, and then you can trace character histories. It's a lot of really useful tools in Mesquite. TNT is how you're calculating your tree. It's a parsimony tool. So I use it mostly for morphology. I don't do a lot of genetic stuff. I don't do any genetic molecular phylogenies. I haven't done it yet, at least. So I do morphology, mostly parsimony. I've tried some Bayesian stuff, but TNT is very good. Mesquite is good. Last time I checked, they were both free. RStudio is good. If you want to do a Bayesian analysis, Mr. Bayes, I know, is popular, and I think it's pretty good. And then as far as data sets, a lot of papers in their supplementary information, if you check, will have Nexus files, which include the scorings and stuff. if you want to use someone else's data, or you can collect your own data, and then they'll include, all the characters, all the taxa, and their scorings for them, usually in a TXT file. So you can check. We do a lot of matrix construction from older phylogenies, so you'll kind of be like, oh, this covers this group, that covers this group. Let's put them together. We'll add some new characters we observed here. Let's add these taxa that weren't in that, and then you build it from the ground up. I've never had to pay for. I don't think. Well, no. I mean, I guess to score stuff, I had to travel, but you can easily construct a phylogeny from the comfort of your home without spending any money, as long as you have like a computer and internet access.
Yeah. Okay. So let's see. This is from Nestle 20, and it's not quite a question, but I think it's an interesting discussion. There is the question why pterosaurs apparently didn't spawn secondarily flightless lineages, but birds did. My guess, this being Nestle 20's guess, is that they were so much adapted for flight that they were unable to. I would also point out, I don't think we can say for certain that there were no flightless pterosaurs, because let's say that one of the places we get a lot of flightless birds today is like little islands out in the middle of nowhere. They're probably not going to leave a fossil record. So there could have been oceanic islands out there with flightless pterosaurs.
It's possible. That's a really cool question. I'm thinking about it. Let's see. So it's possible. So the classic wonderful life idea of punctuated equilibrium, like go back to the Cambrian, where it's like tons of crazy lineages that aren't related to each other. That turned out not to be true, because it turned out to be a lot of the Cambrian stuff is stuff we know, and there seem to be constraints on evolution. The famous quote is like, if you replayed the tape of life, you'd get wildly different things every time, which I say, probably not. It seems that like life keeps doing these, at least animal life, keeps doing like similar things differently and often very strangely, but there seem to be spots they fill. Unless you went back to like the origin of life and life was a silicate-based thing, in which case, who knows? I have no idea. But pterosaurs, back to pterosaurs. It's possible, this is kind of using this constraint of life. Not anything is possible. It is entirely possible that pterosaurs were so adapted for flight that they could not be secondarily flightless. However, I would say there is some evidence of groups that, while almost certainly, while flying, were very comfortable on the ground, like Quetzalcoatlus and that whole group seem to be, or are often reconstructed as being competent terrestrial predators. And some people are like, oh, they're running after prey. I don't know if they're doing that. I know their arms are stitched together by skin, so I'm not totally sure, but we seem to have evidence that they are competent and probably quick relative to a lot of other things, quickly moving on the ground. I don't, I can't say if they're on their way to becoming flightless. They probably weren't. They're still look like they're good flyers, but they were at least playing around on the ground. And to the point of flightless island birds, I think that's a good point. It could be that, we're missing diversity from here or there. The only, actually, no, now that I'm thinking about it, I mean, yeah, they could have existed, and we just don't have samples for them, just because.
It's one of the unfortunate things about paleontology is we just don't know what we don't have, and there's no way to know. Because, like I said, like little oceanic islands pop up here and there, and then they just kind of crumble back into the water over millions of years. And
exactly. What
are you going to do? Look at the bottom of the ocean for a dinosaur bone you have no reason to think is there? No. So all sorts of weird stuff happens on islands, and we'll probably never know what was going on in weird oceanic islands from, the middle of the Jurassic. So who knows?
No.
I mean, you can find old seamounts from what's now the Hawaiian volcano chain going all the way back to Russia. So basically there have been Hawaiian islands, essentially, since the end of the Permian, more or less. So what was happening in Jurassic on what would become Hawaii? Something. But there's no way we're ever going to know. And it was probably weird, because Hawaii is already weird.
Yeah, it'd be incredibly expensive to launch seamount excavations
Yeah, and
I wonder how much sedimentary rock is left on
I would guess very little. I mean, for one thing, Hawaii doesn't put down much sedimentary rock anyway.
No, because in the like Bahamas, you get nice carbonate shelves. But I'm not. That's a continental shelf. It's not a seamount in the middle. Yeah, so I have no idea.
Yeah, that's one of those things where it's just like, it's a mystery. We would probably need a time machine to figure that out. But it would be a very, I'm sure whatever was going on, it was pretty cool.
Oh, no, absolutely.
Islands tend to be pretty cool.
Madagascar was an island during the Cretaceous, and we do know what was going on there a little bit, and it was weird.
Yep.
Big frogs.
Also, Nestlink20 wants me to tell you that you should be very careful while describing Scansoriopterigids as having bat wings, since creationists tend to take that very literally. That's true, actually.
Okay, so
I saw
they have
membranous
membranous patagium supported by an elongate pisiform wrist bone, and then
which actually is not that bat-like if you think about it, because they don't have.
No, it's very not bat-like, but there's some skin in between their fingers too. Right. I guess it's more pterosaur-like than it is bat-like.
Well, actually, I know of at least one creationist who has decided that Yi Chi is in fact a pterosaur, and I was at a loss for words with that one, because I was just like, what? No. The only way you can say that is if you just don't know anything about dinosaurs or pterosaurs, except that pterosaurs have membranous wings. That's all you know about either group. Then sure, you could say that Scansoriopterid. But interestingly enough, an earlier Scansoriopterid that he had been looking at, he just decided it was just a dinosaur. And then he's like, oh, this one, I don't know if it's a bird or a bat or a pterosaur, but I'm going to go pterosaur. And it was painful.
Yeah. I mean, I think one of my so if you're familiar with the kind of fiducia crowd of, I mean, they're not really active anymore at this point. I think everyone's quietly accepted this. But there was a paper that came out some time ago about Scansoriopterids, published by this camp, this intellectual camp that, birds aren't dinosaurs, but there are another kind of archosaurs, et cetera, et cetera. Most manoraptors are secondarily flightless birds. Continued. They published a paper where they're like, Scansoriopterids are the transitional they're not dinosaurs. They're some kind of archosaur that transitioned into birds. And they reconstruct it with these big primary flight feathers on its arm, which aren't preserved.
We don't even have any quill knobs, as far as I know.
Exactly. And then, not long after that, they found Yi and Ambopteryx, which have bat wing. Well, skin attached to 3 fingers and a big wrist element. So, no, and they often that camp of people often criticize people for, it's like you're over reconstructing the feathers. Those aren't feathers; those are collagen. You're making up anatomy.
Oh, I hate the collagen.
They don't, but then they make up primary feathers on the arm, and we find out that there were no birdie primaries. Yeah. It was skin.
Yeah. That is actually one of the things that bugs me, is the collagen in feathers thing. That actually has been gobbled up by a lot of creationist groups. And so one of the things I point out is like, well, collagen isn't known for its melanosome content. But feathers are, and we know that these structures have melanosomes, so...
Yeah, at this point, the criticism of there's no... They're feathers. They're not collagen. There's a ton of work's been done into this. They have keratins. They have melanosomes. However, bizarrely, that collagen argument has been resurrected for pterosaurs by David Unwin. That pterosaurs are naked, and their pycnofibers are frayed collagen. Except for neurognathids, which have keratinous structures on their face, he claims. It was actually, so he published a paper where it's like, pycnofibers are not homologous with feathers. That was the paper. The paper's like, oh, these filamentous structures are not feathers. They're something else. But everyone kind of knew because I think he had pitched it as a poster or something a while ago. And it's like, oh no, they don't have any filaments on their body. They're naked. And he points to like, oh, the wings have skin and the throat pouches and stuff like that. So,
Well, yeah, there were parts like any animal that didn't have. Yeah, that
so
seems like
that paper came out a little while ago.
seems fairly weak to me.
Like I think it came out this month. You can check it out. It's garnered quite a bit of criticism.
No surprise. Oh, so here's one thing about the collagen fibers.
Yeah.
Do you know who Ian Juby is? You probably don't, and you shouldn't. And don't afflict your life with Ian Juby. But do you know who that is?
No, I don't think the name rings a bell, but I'm not sure.
So he's sort of like a second tier creationist guy. Every once in a while, he'll go on with like Answers in Genesis or something. But he's mostly independent. But at one point, he claimed that there were collagen fibers similar to what we see as protofeathers identified from an ichthyosaur. So I traced down this claim. I actually went through and I looked and like, okay, because all he had for it was this picture, which, yeah, great.
Oh, yeah.
Way to cite your sources, guys. Just a picture. So finally, I found the picture, and then I found the actual description of the fossil. And you know what the collagen fibers were? Tool marks.
What?
Tool marks.
Tool mark?
Yeah, from the original preparation in like 1840.
Well, yeah. So in 2003, there was a brief. I think there was something written about monofilament elements in ichthyosaurs that were proposed to be collagen. There's like, oh, they look just like Sinosauropteryx, and it's a 2017 or 2018 paper. And in my opinion, the final nail in the coffin of the collagen thing. That's called "On the Purported Evidence of Collagen in Ichthyosaurs and Bird Feathers," and it's this excellent piece by piece discussion of, it's like, it's not collagen. This is occurring outside of the skin. It's got this. I strongly recommend looking for it. It's by the same authors who did the Sinosauropteryx color reconstruction.
Okay.
And it's a very good paper.
Nice. All right. Well, I think we will get in one more question, unless a super chat comes in, because if you're gonna pay to have your question asked, I feel like it's an obligation to answer it. But let's see. This last question is going to be coming from Vandali 1998, and he asks, if penguins continue their aquatic journey, do you think they will be the first fully swimming dino unless there is already one out there? And I think he just means like sort of like aquatic to the point that whales
they don't
are.
Even come back to land to like lay egg. They reproduce solely in the water.
I think the egg is probably the big thing, because that hard-shelled egg is there's a lot of adaptation towards growing a hard-shelled egg. It has to be exposed to air. But
so I mean, like, speculative evolution is cool.
Mm-hmm.
I love those. Like, there's that book Kong Skull Island where it's like,
oh yeah, that's fun.
It's like isolated, stuff like that. And then there's all those Douglas Dixon ones. I'd say if there was going to be a group of dinosaurs that became entirely marine, it would probably be penguins. Just based on what they're doing currently. That's like was said, the eggs is the tricky one, because at least in like maybe mosasaurs and other marine reptiles, they probably had soft-shelled eggs.
Mm-hmm.
And then boas, I think, technically hatch the soft egg inside of them and then give live birth. So it's possible. I mean, I don't know. I guess I'd need to know more about eggs to know if such a thing were possible. The
tricky thing for me with the hard-shelled dinosaur egg is that one of the reasons that the shell is there is not just structural support, but it also provides gas exchange from the air. Yeah. But it's heavily dependent on being in air and not being covered in water. If you put a dinosaur egg underwater, I mean, for a few seconds, whatever, if you leave it under there, the embryo will drown.
Yeah.
So
no, that's a good point.
That's also one of the reasons why it's hard to retain the bird egg for very long inside the body, is because its gas exchange is primarily coming from the air. And so that, I think, is the trickiest part. You would need some radical changes in the reproductive system of birds. And one of the problems is I'm not sure that most of those things that would lead to being able to be fully aquatic would actually be all that adaptive for penguins as they are now.
Yeah. No, that's a very good
So I'm not saying no. I'm just saying those are the tricks that you have to.
And like almost the exact opposite of like an albatross is like almost an entirely aerial organism.
Yeah, it barely touches the ground.
But it's still got to lay those eggs.
Mm-hmm.
And pterosaurs were probably, at least the big ocean ones were probably, doing a ton of that stuff too, and they still had to lay eggs. Hesperornids, we don't know. I don't think we have any nests or anything, but
it would be hard to imagine them not needing to lay eggs.
Exactly. Interesting, an interesting line of thought, though.
Yeah. Well, I think that we'll wrap it up for questions. And I'm sorry, I know there has been some buffering. Like I said, my internet is doing something stupid. I don't know what it is. So my apologies there. Yeah. So any final words from you, Alex, before I just do some wrap up and channel news and we head out of here?
No, my dog's barking. Stay interested. There's always new stuff coming out. I can tell you from SVP, there's going to be a lot of really interesting stuff coming out. Stuff on dinosaur metabolism. Stuff
Nice.
on new stuff. Stuff. But yeah, plenty of stuff. I'll have some papers coming out. Not one might come out in 2020, depending on how quick.
Nothing comes out in 2020.
Yeah, 1 or 2 in 2021 on stuff people might be rather interested in. Certain taxa that is a little more famous than a lot of the other stuff I work on. But yeah, plenty of things to be excited about. And if you're a person who's into dinosaurs and stuff and vertebrates, it never hurts to stretch your legs and check out invertebrates and Tauponomy, because like I said, I'm someone who doesn't love chemistry, but like learning about how stuff gets preserved is just so cool. And there's a lot to paleontology that's not dinosaurs and even not crocodiles, and it's all interesting and cool.
Awesome.
Even animals.
Awesome. Well, all I have is let's see. Tuesday we are going to have an Eric with Erica. Bent is taking that day off. He has to take a family member to the airport, and so we switched out which days we were going to do Kent with Bent versus Eric with Erica. So if you like Gutsick Gibbon, which why wouldn't you? She's awesome. Come see her and I take apart some Eric Hoven nonsense while we get drunk on air, basically. Let's see. Then Thursday, the Thursday upload is going to be a long one. It's I have compiled all the Fluffy Dino Lads videos into one, and that is going to be premiering Thursday. I am also working on a new, it'll probably be 3 or 4 videos series, and once I have at least all the audio done for that, I'm going to dive right back in and hopefully finish up the rest of the creatures for the Speculative Evolution project. I think we can probably get it done in 1 more video, maybe 2. And yeah, that's pretty much it for the channel news. I hope everyone has a great day. Have a great weekend, and I will talk to you guys on Tuesday. Well, unless, you're a member or a patron, in which case you can always hit me up almost with a pretty good answer time on the member or patron Discord. But, yeah, I'll see you guys later. Thanks for watching, but before you go, I'd like to take a second to thank my patrons and channel members, especially those pledging $20 or above: Ben Hovend, Ian Chen, Sphincter of Doom, Chris Love, Henry Hutanen, and Bob Knob. Their support helps make this channel possible, because as you may know, YouTube is a very volatile platform, and they give this channel much needed additional stability. If you'd like to join the team, a link to my Patreon page can be found in the description, and you can join with the button right below this video on YouTube. Both groups of people get access to my special patron and member only Discord channel, links to new videos before I release them to the public, as well as a pretty direct line to me. They also often are asked to do things like vote on new video topics. If a monthly subscription isn't something that you'd like to do but you'd still like to help out the channel, I also have a Teespring store that has Dapper Dino merch, including mugs, blankets, pillowcases, shirts, all sorts of things. And if none of those things are for you, then please just remember to like this video, subscribe if you haven't already, and share the video. All of those things really do help the channel grow. Well, I'm the Dapper Dinosaur.